In the central dogma, the process by which the DNA is being turned into RNA is called transcription. Once you've made the messenger RNA, when it turned into proteins, that process is called translation. Proteins are complex polymer. The repeating subunits that are found in proteins are called amino acids, and there are essentially 20 different kinds of amino acids. The amino acids have an amine part of the molecule, and they have an acid part of the molecule, and a variable side chain. The variable side chain can consist of acidic residues, basic residues, hydrophobic residues. Based on whether they're charged, like the acids or the bases, or uncharged, or hydrophobic or hydrophilic, the different amino acids has slightly different properties. Each amino acid can hook together by the amino and the acid part of it and form a long chain. The side chain's kind of hanging off of the side. The polypeptide formed can then fold up, sort of like a string of spaghetti, because the polymer part of the chain is very flexible. However, what they like to do is fold up into a ball, so that the hydrophobic parts of the molecule will hide away from the water and the hydrophilic parts will interact with the water. Some of the reactive parts, the acids and the bases, are in pockets on the surface. These typically form what are called the active sites of enzymes. And because those are reactive acids and bases, they can catalyze reactions. For example, when you eat sugar, that goes through a process called glycolysis, which involves a series of proteins that are folded up into a particular shape, such that they recognize glucose. The sugar binds to the enzyme, which is a protein, in what is called a lock and key mechanism. It fits in a very precise way, such that certain chemical moieties are in close contact with certain amino acids that are reactive, typically acids or bases. That allows the enzyme to do a chemical reaction on the sugar, so the sugar begins to be broken down in a series of steps. About 15 steps that break down the sugar allow your body to use the pieces of the sugar to make ATP that fuel chemical reaction of many biological processes in the body. Some part of the sugar will be used to make the amino acids that went into the protein, and some part will be used to make the nucleotides that go together to make the RNA and to make the DNA in the most efficient way. Not all protein are enzymes. Some proteins have a particular shape that lets them fit together, and this forms the cytoskeleton, which gives your cells the shape that they have and allows your cells to fit together to give your body the shape that it has. It's the three-dimensional shape of the proteins that allows them to carry out their functions, whether it's structural, or metabolic, or energy conservation, or information processing. That shape is caused by the amino acids and their chemical properties. But the trick that makes this work, they have to be hooked together in the proper order, one after another. So, a typical protein may have from 100 to maybe 1,000 different amino acids. Where does that order of amino acids come from? It comes from the RNA, and the RNA gets that from the DNA. So what the DNA code does is that the bases in the DNA go together to tell the ribosome through the action of the messenger RNA, what order to hook the amino acids together. This is called the genetic code. So, in your DNA, you've learned there are 4 bases, A, T, G, and C. And the order at which those are found in the DNA, 3 at a time. So, the order of amino acids that are put together in the protein by the ribosome come directly from the information that's stored in your DNA.